Um, Hi, everybody. Good afternoon. I'm Rahat Shabir. I'm recently graduated from Griffith University, and I did PhD under supervision of Professor Changrong Chen. And today I am going to present one of the chapters of my PhD, in which I focused on to the suitability of different biochar as an alternative microbial carriers. Let's start with an introduction. As we all know that rhizobia can fix nitrogen through symbiotic associations, and that can increase um, the nitrogen availability, and that can help to enhance the plant's growth, and can also help to reduce the use of the fertilizers. What's a rhizobial carrier? Usually rhizobial carriers that can carry rhizobia from source to sinks, means from laboratory to the fields. And the rhizobial carrier should have some appropriate characteristics that include the ease of local availability, that should have some appropriate physicochemical characteristics, and they should also support the growth of microbes, and they should be ease of manufacturing and handling of these rhizobial carrier materials. For this PhD project, I worked on the biochar as alternative microbial carriers. Biochars are usually carbonaceous compounds. They can be pyrolyzed at different temperatures, starting from 350 to 100 degree temperature, and they have a very good physicochemical characteristics that could be appropriate for rhizobial survival and growth. For example, they have high carbon content, they have a highly porous structures, and they can retain the nutrients, and they have an appropriate water holding capacity. So these are the important features. So that's why we selected biochars to be used as an alternative microbial carriers. The objectives of my research was to assess the different surface properties and the chemical compositions of the biochars. And then we aim to do incubation experiment in which we put rhizobia into the biochar and store them at two different storage temperatures. That was 28 degrees temperature and 38, to see how this biochar can support the rhizobia even at high temperatures. Furthermore, we assess how different physicochemical properties of the biochar could help or could interact to find the mechanism that how biochars could improve the rhizobial shelf life through structure equation modeling. This is the methodology. I use 6 different feedstocks, and we pyrolyze them at 600 temperature to produce biochar. And then we characterized biochars for different physicochemical properties. And then we used CB1809 strains as rhizobial strains, and we mixed rhizobial strains with biochars using a 50 to 60% moisture, depending upon the water holding capacity of the biochars. And we also sterilize the materials before mixing them with the rhizobia, and then we determine the shelf life after 90 days. Here are some results we got after the physicochemical characterizations of the biochars. And here we divided the physicochemical characteristics into major elements and the relative ratios, and the trace elements, surface morphology, and the key basic properties. And we find that the pinewood biochar have a appropriate total carbon, have a very good calcium, different manganese, and the surface morphology in terms of specific surface area, total pore volume, and the pore size. Then we tested their surface characterization through FTIR analysis and XPS analysis, and we found that different functional groups appeared in different biochars, and specifically XPS analysis was carried out to see how different functional groups could be quantified, and we found that the pinewood biochars have a highly amount of functional groups like ketonic functional groups and carboxylic functional groups that could be suitable for rhizobial carrier. We tested in using the structure equation modeling ahead. And then we have SEM analysis of all biochars, and obvious channels and porous structures could be seen through this SEM analysis. Now we determine the shelf life, and we have the results of the shelf life. You could clearly see that the end of the storage time at 28 degrees centigrade, the different biochars like pine wood, sugarcane, and the oak have a shelf life comparable to the peat. However, at 38 degrees centigrade, we could see that the biochar, pine wood biochar, outperformed the peat and showed the significantly higher amount of shelf life as compared to peat and other biochars. And then we did structure equation modeling. We used 5 or 6 different latent variables, and we found that the functional groups, major elements, and surface morphology have a direct positive impact on the rhizobial shelf life. Yeah, this is conclusion. Different biochars have different physicochemical characteristics, and all could be useful to be used as alternative microbial carriers. However, pine wood biochar, we found that have a most appropriate physicochemical characteristics and shelf life. And among the different physicochemical properties, specific surface area, pore size, pore volume, total carbon, as well as manganese, they showed direct positive impact on the rhizobial survival rate and shelf life. Thank you.